On behalf of the Department of Ecology and Evolutionary Biology, it's my pleasure to welcome you to the 15th Annual Early Career Scientist Symposium. And we are all very excited with the program that we're about to hear today. Our theme for this year is stable isotopes, as you all know. That's why you're here. So we have 8 wonderful speakers who are all showing where the frontiers of work in stable isotopes lie today. And the talks are going to cover a variety of ecosystems, isotope systems, and conceptual topics, including aquatic and terrestrial settings, paleontological, archaeological, and modern records, multiple isotope systems, microbes, plants, and animals. So we're trying to be very inclusive with the range of topics that we've selected as speakers. And then the poster session, which will be for an hour in the afternoon during a long coffee break, is also going to feature an additional set of topics, especially by the folks here at the University of Michigan who are conducting research in stable isotopes. So I'd like to start today initially with just a few thanks. And of course, the first thanks need to go to our benefactor. This is Nancy Walls, who's an alumna of the university. She was a pioneering microbiologist at Georgia Tech, and she left a bequest that has, to us, to our department, that has funded these early career scientist symposiums over their entire existence. And so we are very grateful to her and her family for this bequest. Next, I'd like to recognize the members of the organizing committee, whom you will see throughout the day because they are introducing the various speakers and helping out with all of the logistics of the program. And in addition, I'd like to acknowledge these 4 folks who have done all the work behind the scenes to create the wonderful breakfast, the coffee breaks, the meals, and the registration, and the program, and the beautiful graphics. So thanks to all of these people for making this happen. So I'd like to just give a brief introduction, excuse me, particularly to those of you who do not work in stable isotope systems, about why we are here to look at the breadth of topics today by giving just a few examples of applications across environmental sciences. And by this, I'm referring essentially to earth sciences, ecology, evolution, and also anthropology. And this is what got me so excited about stable isotopes when I was a graduate student, and why I've continued to collaborate with geochemists who are working on these and to use some of these applications in my own research. So I want to give about 7 brief examples to show the breadth of stable isotope work. And here, starting with, deep time, is what those of us in the earth sciences call the Zachos Curve, named for Jim Zachos, who used to be a postdoc here in earth sciences. And what we're looking at here is Earth's time, geologic time on the horizontal axis, older to younger. And we're looking at the oxygen isotope record from forams that live in the deep ocean. So this gives an averaged ocean mean temperature, and it allows us to see that over the last 65 million years, we've had a very substantial changes in Earth temperature over time, and it puts into context the temperature changes and the climate changes we're having today. For example, we are here, of course, but actually our global warming has risen considerably, and what we may be looking at are times that are back to the Eocene, and very quickly. And so these are insights that we can get from the oxygen isotope record. From my own fossil sequence that I've worked on for many years, decades really, in the Miocene of Pakistan. Now we have another stable isotope record, and here we're looking at the carbon record, and here is the scale of carbon with more depleted, more enriched values, and here's geologic time. And so we have this amazing fossil sequence there that's about 18 million years long, and it's been sampled over time for 2 different kinds of records. The brown points represent samples from ancient soils, and the green diamonds represent enamel from mammalian herbivores, mostly ungulates. And what they show us is that during this long record, there was a huge shift in carbon isotopes in both the soils and the mammals, and they reflect a transition from vegetation that was dominated by C3 plants to vegetation dominated by C4 plants. This was the first fossil record to document the expansion of C4 grasslands back in the Miocene. And we'll hear more about this system this afternoon from Alison Karp when she provides insights from completely different sources of information. So these are insights, two examples of insights from the fossil record. Now, C4 plants represent one of 3 photosynthetic pathways that plants exhibit. And this work here, although we're looking at a phylogeny, what I want you to notice, and this is a phylogeny for grasses, the family Poaceae. And this family has 2 different photosynthetic pathways present. And the blue lines here represent the clades in which C4 photosynthesis has arisen. And it shows that C4 photosynthesis arose independently several times in this family, but those are nonetheless those evolutionary transitions are clustered in the phylogeny. And the reason for so this is a link between stable isotopes and the evolution of this very diverse group today that is very dominant in many ecosystems. And C4 and C3 plants, as we will see, have very different, distinct isotope signatures in carbon space. So another connection to evolutionary biology. There have been a lot of applications of stable isotopes in food web analysis and diet. And here's one that is somewhat interesting, looking at condors from North America. And so what we're looking here at nitrogen and carbon space. And what we're seeing is gradients from terrestrial to marine and from C3 to C4 plants in both carbon and nitrogen data. And from bone collagen of subfossil and modern condors, we can see that during the late glacial stages, the Pleistocene condors appeared to be consuming a lot more of a marine sources, particularly whales and seals that might have that they may have scavenged on beaches. Later, the condors that existed as wild populations in Western North America were eating primarily terrestrial herbivores, as they were particularly those that were located more in the interior of the country. And of course, they've undergone a huge bottleneck, so condors are highly endangered. And the few remaining ones now have a very different isotope signature. They mostly live in the Southwest, and they're supplemented with carcasses of cattle from feedlots. So now they're showing more of a C4 signal in carbon space because of what the cattle are being fed. So just to show it, this is simply an example of changing diets for a set of very interesting birds through time. Another very interesting example that's relevant to forensics and conservation also again involves carbon, and where the carbon scale is on this axis. It's carbon and oxygen. And this is an example in which there were confiscated carved ivories taken from the Kenya airport. And they were analyzed for their stable isotopes of carbon and oxygen. And the confiscated samples are in these bright red squares here, and they're being compared to a range of values of elephant ivory in Kenya, hippopotamus ivory in Kenya, and in fact, most of the confiscated ivories have a signature from Central Africa, where the hunting of elephants is illegal. So this was one way of determining that a lot of these ivories were not from legal sources at the time. And there have been other similar applications, many interesting applications of isotopes in forensics. Finally, a couple of last examples. So far, we've been looking at examples in which the stable isotopes are looking for, if they're looking at organisms, are looking at some kind of a mean value over the entire year or for longer time average periods. But another very important aspect, and this comes from the work of Dan Fisher's group here, is to look at, in this case, changes in oxygen isotopes within individual growth rings of mammoth and mastodon tusks. And this gives us not information about the seasonality of the environment in which these animals lived, and it also tells us something, in this case, about the timing of the season of death of these animals, and so this gives us tremendous insights into life history and how those life histories changed over the period when these animals were disappearing from the earth. And the last example, which always makes me chuckle a little bit, is getting even finer time resolution, is looking at carbon isotopes on individual hairs from humans who are traveling to different parts of the world. And here we're looking at an individual who moves from the US to the UK, and then someone who moves from Europe to the United States. And in each case, the folks who are within a few months, the hair records that those who are living in the US are consuming diets that are dominated by C4 plants, namely cane sugar and high fructose corn syrup. And in fact, other similar studies have shown that these carbon isotopes in the average American hair has more C4 signal than that of Mexicans because of the prevalence of high fructose corn syrup in our diets. So very interesting the way it's giving insights into the food system. So finally, my last remarks are, if you look at this background picture, this is one last interesting aspect of isotopes. This is an isoscape. That is to say, this gradient of color across an enormous region, in this case of isotopes of heavy hydrogen, from hair, again from hair. And this is also a way of showing the ways in which the physical processes are fractionating these isotopes across huge geographic regions, and then the way the plants and animals reflect this. So with that, I'd like to get us launched into the program. I'll just remind everybody that the poster session should be very interesting this afternoon, so I hope you're able to stick around for that. And now I'd like to invite Ben to introduce our first speaker.